yeah, I think that might be her right now. Yep. Okay, fantastic. Okay, we can properly get going. All right. Welcome. Thank you all for showing up. This is meant to be a study session in the sense that I hope to learn from you how to improve this section of our roadmap and update about success criteria and related matters. And as I mentioned, there are sort of associated topics in there, such as metrics and constraints and scale separation. And that means that I actually have quite a few slides because I decided to, okay, the recording started. Excellent. I decided to just try to throw everything in, put everything on the table that is currently in the draft that we're working on. And so even though we have a 2-hour session set aside, I think it's quite possible that we can't get all the way through. In fact, I think just based on looking at what it takes to go through some of the slides, my feeling is halfway is very likely. But I hope that we can make serious inroads anyway. And as far as doing the whole thing is concerned, I'm happy to do a second session another time. But if you want, if most of the people here feel like it, I can also just keep going longer. I don't have to be anywhere really soon. If we do another session, then it's not going to be until 2 weeks from now because I have another thing going on the following weekend and in between, so I won't be here. Okay, anyways, that's enough about that. So here's an outline. The first 5 parts are the draft content, the ones on the left there. The points on the right are discussions that I hope we can have. So first I'll introduce the topic of success criteria for whole brain emulation. Then proposals for the discovery of success criteria and those that we managed to collect and categorize so far. Thirdly, associating success criteria and specific metrics. Fourth, an important topic about system constraints. And finally, integrating the proceeding with the question of scale separation in whole brain emulation. And the proposed discussions are about each of those steps. And then perhaps we can briefly address the questions of what is now being called lo-fi whole brain emulation and see how that, how what we're talking about here informs that. Okay, so let's start by defining this section, the success criteria section of the roadmap. Beginning with some expressions of the foundational principles of brain emulation. The Church-Turing thesis posits that any algorithmically solvable function can be computed by a Turing machine, i.e. a computer. If brain function can be described mathematically, then its emulation on a computer should be possible in principle. The Kullback-Leibler divergence is an expression that gives insight into the difference between an attempted emulation and the original system in terms of the distribution of probabilities in the original P and the emulated system Q. And you can interpret the metric as measuring the loss of information by emulation. And emulation where that loss is smaller has greater fidelity. While most popular, those are not the only ways that you could describe or evaluate emulation mathematically, and I briefly note a few other interesting theses with relevance to emulation. Stephen Wolfram's computational irreducibility refers to systems so complex that the only way to predict their outcomes is to simulate their computations, and it's assumed that the original algorithm implements the shortest path to the derivation, and any other path is less efficient. Gödel's incompleteness theorem is a bit of a different beast, relating to emulation largely in terms of the hurdles it proposes. Essentially, applied to cognition, the theorem applies that any system of thought, oh, there's somebody trying to get in. Any system of thought, including the human mind, represented as a formal system with consistent rules will inevitably encounter true statements that cannot be proven within that system. And that's only a problem if humans nonetheless possess a capability to understand and reason beyond those limitations, allowing them to grasp concepts that algorithms could never fully process or prove. I don't think that most of us worry seriously about it, irrespective of Roger Penrose's position. I also mentioned the Kantorovich-Wasserstein metric, which I also have a quick little overlay here for, because it offers another metric for the cost of emulating one system in the expressions of another. But I don't think we actually really need to get into that at the moment. Trying to be thorough the first time around, let's further ground what we're trying to do in some philosophy of personal identity. And we could draw on innumerable philosophical stances. For the purposes of what follows, I'm naming just 3 here and presenting their conclusions about a personal identity in an order that builds up the argument that the nature of personal identity permits us to use what ought to be preservable in a connectome as the source of a brain emulation that can be claimed to enable survival of a person. After all, that's one of the reasons, though absolutely not the only one, why we seek to achieve whole brain emulation of a certain kind. If you read Locke's elaborate series of arguments, you find that he derives the conclusion that the self persists through memory, and he ties that into consciousness and persistence of memory into distinguishing oneself from other thinking things. And Locke even specifically describes that persistence even if the material substance of a person changes. And that was, hundreds of years ago. Note that what I mean when I write memories is not just snapshots of episodic memory or images of people you know. It refers to the way in which synaptic weights determine patterns of processing, and that includes characteristic behavior, cognitive processes that produce a person's characteristic conscious awareness, even qualia, if you like. Next, I quote Hume here for his realization that self is a generated thing, like a perception composed of multiple contributions and not fixed over time. Hume already proposed that therefore a unitary or static impression of self is a kind of illusion. And if I'd chosen to proceed to philosophers who are active in neuroscience, I could have included Thomas Metzinger here for his very detailed ideas about his self-model of subjectivity, which includes both the generation of a virtual model of the world and identification of a self within that. But I think Parfit quite possibly discusses personal identity in the manner that is most explicitly and most closely related to brain emulation and even its application to mind uploading. According to Parfit, it doesn't really matter if you slowly fly to Mars in a rocket or if you use a teleporter to reconstruct yourself there using a different set of atoms. It's always a new you, and survival is achieved through the psychological connectedness before and after. Because it's always a new you, survival must be a matter of degrees. Today, and also, of course, when a patient has undergone whole brain emulation. Mirinda, who leads our ethics framework project, is drafting a set of foundational papers on the philosophical underpinnings of whole brain emulation. So perhaps we can have an entire separate session about that as those drafts come along. Based on the preceding, we can posit that successful whole brain emulation is ideally a process that can be used to achieve survival to a satisfactory degree by the standards of a person subjected to the procedure. Of course, different persons are likely to have different standards. For the sake of a roadmap and for the science, we can aim for standards that represent a consensus of sorts. Alternatively, or in addition, we could attempt to aim for objective standards wherever those might be identifiable. And furthermore, we can open up the scope of our set of goals to include a few that express additional strong reasons to seek to achieve whole brain emulation, such as whole brain emulation in the service of brain science to vastly improve our understanding of brain, mind, thought, etc., or whole brain emulation used to discover how the mind is doing things that AI so far is incapable of, or, and I personally find this even more compelling than the application of whole brain emulation to personal extended survival, that is to seek to achieve whole brain emulation as a path to human substrate independence, which may very well be an essential prerequisite for mature and advanced intelligent species. In that case, whole brain emulation has value even in the absence of the survival of any one particular individual who presently exists in biological form and doesn't have the benefit of substrate independence. Of course, we can use a similar consensus defining process to determine the principal set of goals themselves if we want to. Let's say that our goal includes the desire to make sure that our model allows us to study mechanisms of disease or the effect of drugs where they might manipulate molecular processes. In that case, our model of synaptic change might be one that includes details of the process of long-term potentiation. Letting someone in. Calcium ions in that case, activating enzymes that subsequently modify the spatial conformation of molecules, which results in greater insertion of AMPA receptors into the membrane and all of that. Alternatively, let's say that our goals are restricted to certain aspects of cognitive information processing, for example, in the visual system or episodic memory. If we merely wish to tune synaptic strengths, but not to affect that process as in the previous goal expression, then we might choose to stick with spike timing dependent potentiation on synapses between spiking neurons, shown here in B and Poo's famous graph. Of course, this is a really simplistic example of the relationship between goals and success criteria, because if that cognitive processing is part of a whole brain emulation, you might very well demand that modulators such as drugs should affect cognitive processing as well. Now, the success criteria we choose will strongly influence which kinds of goals can be achieved. For example, imagine that we decided that only the external I/O behavior of a system matters. This is the position taken in black box system identification, such as when you don't use the internal architecture or composition of a system to inform the model that approximates it. If the model is constructed by deriving its transfer functions from observed input and output, then the functions implemented will depend entirely on the observed activity, obviously. Those might be many hours of recordings, but except in cases of very small and short-lived organisms, they're unlikely to include an entire lifespan. And even then, there may be implicit transformations never observed, but supported by the functional architecture of the brain in question. Consequently, that approach is not likely to replicate some of the possible I/O behavior, and importantly, it doesn't guarantee that any internal conditions are implemented in a manner that would produce similar subjective experiences. There are plenty of examples where you quickly run into this problem. For example, our journal club about the Lapalainen et al. Nature paper on the fruit fly visual system model that applied a model implementing stereotyped convolutional copies of an average connectome constraint circuit. That was the example we looked at 2 weeks ago. So the success criteria included mechanistic predictions within a simple visual task there, but wouldn't satisfy success criteria about the uniqueness of an individual fly visual system, nor would that approach necessarily reproduce implicit functionality that's applicable to visual tasks outside of the ones that the model was optimized for. Another example is the multi-input, multi-output system identification method used to build a hippocampal model based on merely 64 recordings in each of CA3 and CA1. And lastly, we can apply the same black box system identification concern to proposed lo-fi whole brain emulation procedures that rely on external observations only. They're unlikely to reproduce subjective experience or the realistic evolution of such experiences over time. Determining the success criteria for whole brain emulation is not merely important for a particular experiment or emulation attempt. It stands to reason that recognized success criteria can determine the course of development of an entire field of science. I added the tongue-in-cheek example here, pointing out that excellent progress when the success criterion is a nuclear explosion doesn't necessarily effectively move forward science and technology towards fusion power. The following points show how technological and scientific effort is guided by success criteria. Success criteria determine if a specified goal has been achieved. A set of validation metrics will determine if success criteria have been met. Those validation metrics can only work if certain kinds of data are available. And those requirements demand that the development of certain kinds of data acquisition methods and data processing methods come along. So you can see how progress in the field and what that progress constitutes may be strongly affected or directed by a concrete perception of success criteria. One more point is to note that explicitly that using the evaluation of methods in the context of success criteria is a way to prune the various paths in a roadmap. And we tried to do a little bit of that in some way 2 weeks ago when we were digging into that Nature paper about the connectome constrained D&M approaches, for example. All right. Now let's look specifically at the potential success criteria for whole brain emulation. Oops, there we go. How might we determine success criteria? Let's consider this in general. A top-level framework might be to clearly define the purpose of the fields, then to understand constraints of the system we're studying, to set benchmarks and tests, and to learn from stakeholders about their needs and requirements, not necessarily in that order. It's just a sort of very quick overview. Now, being systematic, detailing a little bit more what this general process might look like, as one might describe that in a roadmap document, because obviously that's what we're trying to work on here. Clarify the problem being addressed as well as the mission. What's the ultimate purpose of the science or technology? And this is typically tied to the problem the technology or scientific inquiry is addressing. In the case of whole brain emulation, the goal might be to recreate human cognition in a synthetic medium, for instance. Contextualize the problem, understand the broader context. For science, it might involve understanding a natural phenomenon, while for technology, it's often solving a practical problem. The goal of artificial intelligence, for instance, as an example, might be to create systems that can perform human-like tasks autonomously. Determine essential functions, break down what the science or technology is meant to do. Each core function could act as an independent success criterion. In neuroscience, a success criterion for brain mapping might be accurate capture of neural network structures, for instance. Establish minimum functionality. For any science or technology to be successful, there must be a minimum viable level of functionality or accuracy. What features must work for it to be considered operational or useful is something you might ask yourself. A drug must show, for instance, statistically significant improvement in patient outcomes to be deemed successful. Success criteria must include measurable outcomes. These can be performance benchmarks, accuracy thresholds, reproducibility rates, or other quantifiable results. In space exploration, for instance, a success criterion for a spacecraft could be a specific orbit achieved, or in gene editing, the accuracy of targeted edits. For sciences that involve modeling or simulation, like whole brain emulation or climate modeling, fidelity to the real-world system is a common metric of success. This includes the level of resolution needed for the model to be considered accurate. Success criteria should be grounded in what's realistically achievable, given current or near future technology and knowledge. The success benchmarks should stretch the limits of innovation, but remain plausible. In renewable energy, a success criterion might be, say, a solar panel with 40% efficiency, based on realistic projections of material science. And in fields with significant complexity or uncertainty, success might be incremental. You should set short-term goals or milestones that track progress towards the ultimate goal. So for AI, for instance, an early success criteria can be to focus on narrow tasks like chess, with long-term criteria for general intelligence. Consider the needs and expectations of those who will use or be affected by the science or technology. So success criteria should reflect practical usability, ethics, societal acceptance, all of that. In medical research, success criteria are often patient safety and regulatory approval, as well as therapeutic efficiency, for instance. Success criteria should account for the robustness of a science or technology. How well does it perform under different conditions or environments? Can it handle variability and unexpected challenges? In climate science, models that can predict global warming under a range of scenarios and data inputs are considered more successful. Prepare for cases where initial criteria are not met and include fallback options or alternative paths to success. Success criteria should include how well the technology or scientific model performs in a real-world scenario or simulations that faithfully reflect the real world. So for instance, for an autonomous vehicle, real-world success criteria would involve navigating varied traffic conditions safely beyond simulations. In the context of scientific research, a key success criterion is reproducibility. Can the results be independently replicated by others? Will the science or technology continue to perform over time or under different circumstances? That's crucial for ensuring the success criteria are not just short-term metrics. Can the technology or scientific method scale from small trials or pilot programs to widespread use? Particularly in science and technology development, initial success criteria may evolve as new data or breakthroughs are made, so you have to regularly revisit the criteria and adjust them to new capabilities or understanding. Okay, this one here, I think this should be obvious, but note the possibility that we might want to entertain goal variants with somewhat different success criteria. That's an option. Here's the process that we decided to use to build our first set of success criteria for whole brain emulation. First, we identified a fairly large batch of possible candidate criteria without imposing too many scope constraints. Then we categorized the candidates and reduced them, especially where there was a lot of similarity or overlap. We applied a set of evaluation judgments until we had a fairly stable resulting set. Once there, we can proceed to deriving metrics that allow us to test if success criteria are met, and then, of course, to apply that to attempted emulations. And obviously, there's no way to do that perfectly the first time around, so it has to be an iterative process. Pooling results, when you have your criteria and metrics, it's possible to think concretely about the quality of an emulation, much as you might score the performance of a machine learning algorithm. Now, many of the same things that you would worry about in machine learning also apply there. So for example, you would worry about generalizability, ensuring that the collection of tests ultimately identifies methods that are truly improving progress towards the goal and not simply becoming optimizations on one or a few metrics that you might see, for instance, when there's a particular kind of visual task that is always being used as the metric for, say, some machine learning on vision. Still, none of that's surprising, and it's clearly better to have expressed success criteria and metrics than to just rely on hand-waving claims. If anybody is curious about explicitly using a machine learning mindset in neuroscience, then I would suggest, for instance, the Neuromatch Academy, co-founded by Cornell Cording. That's a great place to learn about that. Connectomics is certainly not a solved field, but a lot of progress has been made, and I'm really glad to hear about the additional work that's just about to come out. Thank you there, Keith. But yeah, so there's been a lot of progress, at least in the first stages, I would say, of that progress. So alignment, segmentation, 3D reconstruction, cell identification, synapse finding, all of that is now supported by relatively useful automation. Given the large number of components, human proofreading of even a small percentage of that is still definitely an issue. But the real problem is clearly on the side of functionalization. There have been examples of functionalized single neuron models, for instance, those that were developed by the Blue Brain Project and the related Human Brain Project output. That's not the only one, but that's, a very big resource that has those. And we've seen work by, for instance, Xu et al., Philip Xu, and the recent Lapalainen et al. Paper from the Cording Lab. Those are some examples of attempting to do functionalizations of some sort, but there are unanswered basic questions lacking clarity of judgment. If you know what you're aiming for, then you can determine whether applying one modeling method over another gives you any relevant improved performance, and you can compare that with the cost difference. Apply any number of optimization techniques, deep learning, Bayesian, statistical regression, what have you. The toolbox is obviously pretty deep, but as always in modeling, the most important thing is to understand what you're trying to accomplish, not to randomly pick a tool. Now, before I show you the spreadsheet of candidate success criteria, a few more words about considerations that went into the process. An important thing to realize is that at the highest level, deciding goals, picking a desired outcome, is not by itself an objective or technical process. Rather, we start with fairly subjective desires from which technical requirements are derived. An example here is that you might wish an emulation should preserve long-term memories at least to the degree that they're preserved in a typical adult of age 60, and you might wish to guarantee reliable retrieval of those memories with at least the precision requisite queuing and ease of, say, a 30-year-old person. There will be some notable goals that are highly specific to the reasons why we care about high-fidelity whole brain emulation in the first place. For instance, we care specifically about the ability to recreate characteristics that are unique to one individual, and that has an immediate impact on milestones and techniques. For example, when you may or may not find that producing stereotypical neural circuits, like in that paper from 2 weeks ago, is acceptable and satisfactory. Let's keep this simple memory example going just a little bit further to outline the approach our survey study took. We expressed the high-level desire, and here are some ways that this can be translated into more technical and measurable requirements. So an ensuing requirement could be that there's some specific maximum acceptable degradation of neural tissue as a consequence of sample preparation. There might be a minimum degree of molecular tagging required if our criteria can only be achieved by aiding the microscopy interpretation process that way. And there is likely some minimum spatial resolution, some minimum acceptable imaging error rate. There's probably some minimum size of data sets used for validation, for example, memory queues that are established in advance. And it will be important that the process used to identify system properties and to translate collected data into model parameters is technically feasible, and that the result is of a kind where established validation metrics that correctly evaluate meeting success criteria can be used. And as a last example, it's probably necessary to go beyond the memory retrieval test and to also confirm that component functions of the model present a relatively good approximation of biological component functions to the extent where that improves the likelihood that matching circuit behavior goes beyond the limits of observed validation data. We're almost at the spreadsheet now. A quick reminder of the parts of our study process that led to the resulting draft set of proposed success criteria is this. We used survey opportunities with stakeholders to sample their expression of candidate success criteria, and we didn't really put any constraints on them when we did that. We then categorized what we collected so that we can understand at which milestone on the way to human whole brain emulation candidate success criteria might apply. Further, we made an effort to reduce the set of candidates by acknowledging when testing if a success criterion might be met is hard or impossible to do objectively. Aside from doing more surveys and further processing the results, there's still work to be done to determine what constitutes a consensus most useful to the goals of this new field of study. Or alternatively, to match subsets of success criteria to sensible milestones, such as in silico work, work in C. elegans or fruit flies, and other candidate organisms, partial neuroprosthesis, and so forth. We started to produce validation metrics associated with a subset of success criteria where that fits into the carbon copies project on the in silico brain emulation challenge. But many more validation procedures do remain to be proposed. And data sets of the type and precision usable with those proposals have to be identified or generated where they aren't already available. In our brain emulation challenge project, we've also drafted a concrete testing and evaluation process, but there as well, a lot remains to be done. It was good to see in that Nature paper from 2 weeks ago that the authors clearly spelled out a proposed purpose and their method of evaluation. But of course, that wasn't obviously or easily connected with its place in a path towards whole brain emulation, which wasn't necessarily their ultimate goal anyway, because overlap with success criteria for whole brain emulation has yet to be something that can be established rigorously. All right, in a moment, I'll open up that spreadsheet to offer some concrete insight. You're very welcome to explore it yourself and to help us improve the survey and its post-processing. There's a discussion part of this session where I'm going to share the URL for that spreadsheet in chat. The proposals here were collected from 3 sources. 2 of those were surveys conducted specifically with the roadmap update in mind, and the third source is the 2008 roadmap document that was published by Sandberg and Bostrom. And you'll see that candidate success criteria proposed by participating stakeholders tend to express cognitive and experiential goals. In total, we collected 59 candidate success criteria. That was after getting rid of duplicates, and we grouped those into 19 categories belonging to 7 high-level groups. Let's see if this works out nicely. Yeah, here we go. Okay. Down below, I'm not sure that this is showing up on your screen, but there are 4 sheets here that are relevant to the discussion right now. The first 3 each supply proposed candidate success criteria and 1 for each source, so each of the survey events and the roadmap. And the 4th sheet is where we carried out categorizing, reduction, and some further evaluation. And like I said, you'll have the opportunity during our discussion to browse through them in detail. So I won't get into too much detail right now, but I'm going to point out a few things just to give you the gist of what's going on here. Let's see here if I can read that properly. So as you can see, we allowed the criteria to be expressed in whichever way best suited a participant. So for example, I am dynamically evolving, not a static snapshot, have plasticity comparable to now, or avoiding dissociative disorders, the ability to associate yourself with past memories, ownership of your mental states. Or let's see here, picking something from the second sheet, cognitive skills at least as good as before, or let's see, same personality traits on standard personality tests within some limit typical of normal change over time. Some of you who participated might recognize yourself as the author of one of these, or recreation of neural patterns or propagation, rhythm of activity in various modes or states of the brain, such as sleep, unconscious, etc. And let's see, one more. Let's go for the bottom one. Know my boundaries, embodiment. And then from the 2008 roadmap, we've got the same outward behavior and results, a one-to-one model where all relevant properties exist, behave within the range of what one would expect from the original, detailed and correct enough to produce the phenomenological effects of a mind, and finally, emulates a particular mind. You can see that these get into all sorts of concerns that stakeholders have when they consider emulation, and especially when they consider long-term goals where it's used as a part of a medical procedure with patients. Now, our attempt to make sense of that collection. Okay. You can see from the columns up here that this was a process of grouping and merging to a non-redundant collection. Each of the unique candidate success criteria was given an ID to make subsequent references a bit easier. And you can see that those IDs, like P1, for instance, they relate back here to say, here it says psychology. They relate back to the high-level groups that we identified. There are several categories in each group, and I'll get back to that on the next slide when I get back to the presentation. In columns F to M, which I don't think are visible right now, you can see that we were aiming to find subsets that are of near-term importance, for example, for our in silico project. And I'll say a bit more about that as well. So let's get back to these slides here. And like I said, I'm going to share the presentation, the slide, the spreadsheet with you. And so you'll have every opportunity to go through that in detail. Here are the high-level groups shown in different colors and the categories within them. Some proposed candidates turned out to be about clarifying definitions more than anything else. That's this little group up here. Some proposed candidates turned out to be about, the largest collections, this one here and this one here. These were either about the psychological nature or they referred to cognitive skills. Then neural circuit criteria are, of course, of immediately applicable relevance. And then there were embodiment criteria, societal criteria, and some that were quite different, such as about acceptable medical whole brain emulation protocols, about the development process, or of a legal or regulatory nature. As I mentioned, aside from using the study to support work on the updated roadmap, we were also looking for criteria that are grounded in whole brain emulation goals that we could immediately put to work in our brain emulation challenge project. So we needed to consider which ones could be tested through suitably objective measures, and that they did not depend on, say, the inquiry and response with a human patient. And we considered how to apply them or reformulate them within an in silico ground truth examples kind of case. Some additional limitations applied to proof of concept tests, where the scale of example systems would be fairly small, and we wanted to know which ones would serve well in examples that focus on the functionalization challenge that we want to demonstrate. I've already mentioned this, just writing out those limitations again. And of course, we really want to make sure that the subset we use, even if reformulated, still fits squarely within the overall goals of whole brain emulation that are described by a complete set of consensus success criteria, so that the in silico effort doesn't lead to a dead end. Now, we'll get to a summary of first results. We have 25 of the success criteria that met the requirements I just listed, and we added the limitations of working with a very small example system. And then, of course, at this stage, we're not too worried about any criteria that address wishes for extensions beyond normal brain function. So at that point, we're left with a subset of 12 candidates. And here they are. Observable behavior and responses are sufficiently similar. Mental performance is at least as fast. There is dynamic evolution through plasticity. A specified set of memories possibly encoded specifically for the test is retrieved sufficiently well. Objectively verified viable memories are retrieved with a sufficiently similar set of correct and incorrect answers. The emulation is able to form new memories and learn new skills at least as well as before. Reconstruction of neuronal circuits through system identification and tuning of properties is sufficiently accurate. Multi-input, multi-output neural traces match within a specified envelope. Spatiotemporal patterns, brain rhythms, synchronization are sufficiently similar when stimulated. Spatiotemporal patterns, brain rhythms, and synchronization are sufficiently similar in specific brain states or modes. Sensory input for sensory experience is provided, and the emulation is able to act or interact, i.e. affecting its world. We reduced the set one last time because for our challenge project, we really wanted to focus on the translation or functionalization problem, not on the performance of a chosen implementation of models. And with this, we were left with 8 of the expressed success criteria. And as expected, you can see that the neural circuit criteria take up a much larger proportion in this subset now. Okay, success criteria are only useful if you can test if they've been met. So let's talk about metrics. This is really the second main topic in here. I think this diagram of a putative protocol for the use of metrics in a process of model improvement will not be particularly surprising to anybody. Not shown here is the process of data collection and initial reconstruction steps, because it's really focusing on the functionalization or translation problem. We can discuss diagramming at a later stage of our roadmap effort, so I'm just going to skip that for now. A quick aside about the appropriateness of thinking in terms very similar to machine learning problem. I posit that cognition is about information processing. That's not just me positing that, obviously. We already saw that what Church-Turing says about using Turing machines to implement or simulate mathematical or algorithmic operations. The machine learning problem at heart faces the same challenges that we have in brain emulation, namely the problem of appropriate model selection and the problem of finding the best parameter estimates within the model constructed. As in every machine learning problem, you have to pay attention to both verification and validation, where verification is about ensuring that the implementation of a model is well done, and validation is exactly about meeting performance success criteria. Even though every modeler should already know this, I feel I have to mention the curse of dimensionality and the associated problem of combinatorial explosion. Both are exponential problems and are caused by the number of operational components and their number of possible interactions, particularly if your modeling or machine learning problem takes a brute force approach. Illustrated here for some randomly selected modeling resolution, not only is the optimization problem large, it may also have a lot of local optima. For this reason, the emphasis on reducing the problem size, like in that Lapalainen paper we read 2 weeks ago, is a valuable case study. I've mentioned on many occasions that I'm convinced that the question of computational cost in whole brain emulation is not at all in the performance of the final implementation of an emulation, nor is it in data collection, data post-processing, or the early stages of reconstruction and identification. Almost all of the computational burden lies in system identification and the translation of data to acceptable parameter estimates. If that isn't immediately obvious, consider that you'll probably have to generate a batch of candidate emulations, and that to optimize and validate each emulation in the batch, you have to run each one anyway, many times over. So it's like a multiple of the problem of the implementation ultimately anyway. A good validation protocol for whole brain emulation will probably require validation at multiple scales. Examples are at the cellular level with firing pattern, stimulus response, and synaptic plasticity, or at the circuit or network level concerning connectivity, signal propagation, and consequent emergent properties, and the behavioral or cognitive levels where we might evaluate sensory processing, decision-making, motor output, and learning, just to name a few. Okay, I think I basically already mentioned that. And this as well. Maybe I should have been a little slower there. Aside from scale, metrics also apply to different sorts of measurements. You might validate structural accuracy by measurements of neuronal morphology or of network connectivity or 3D reconstructions. You might validate dynamic similarity by measuring patterns of firing or timing characteristics of synaptic transmission, or you might validate functional output by measuring behavioral responses or decisions. And you might validate cognitive equivalence by measuring memory performance or test reasoning or something like that. I already mentioned that the process is undoubtedly iterative and will involve model batches. Now, while having to make batches and iterate increases the amount of work, I think that validation at multiple levels can actually lead to shortcuts, improved outcomes, and a reduction of the overall work. The Brain Emulation Challenge platform that we've been working on is actually intended to help researchers to develop and test these processes in search of best practices. Let me mention a few additional challenges. If validation depends on in vivo data from unique individuals or organisms, it may be impossible to collect enough data. The technology needed might not exist or may not exist for the organism in question, like in the case of, say, having good gene lines. In human volunteer patient protocols, there are very serious limitations on the kinds of procedures that are possible that you're allowed to do. If we have to guarantee correct identification or correct modeling of every cell or synapse, or in other cases, there might be missing knowledge, the resolution or temporal scope of the validation protocol may not pick up on problems that only exhibit over large time spans, such as an undesirable evolution of a plastic system. And of course, our understanding of the success criteria itself, those might be insufficient. The next few slides are illustrative of the state of the art in validation methods and metrics in the computational neuroscience that may apply to brain emulation. You can have the slides as well, just like you can have the spreadsheets, and you can read them at your leisure. So in the interest of time, I won't walk through each one in detail. Let me just mention here that we did indeed choose to include graph theoretic metrics as a part of the validation process used in our Brain Emulation Challenge platform. as well as examples of each of the metrics listed on this slide. We haven't implemented as many functional validation metrics yet, focusing mainly on I/O fidelity right now. I'm not going to talk too much about all of this, as I said, so I'm just going to carry on. Perturbation studies are actually, or maybe sensitivity analysis are useful and applicable both at the organism level and with smaller in silico systems. Okay, causality and perturbation tests comes up again here. So we did some rudimentary causal interventions in the case of our in silico experiments, performing lesions as examples of incomplete emulations as a part of a limited test of the evaluation reporting. And as we already saw 2 weeks ago, when we looked at the Lapalainen et al., that's an example of using deep learning, for example, to optimize parameters of a piecewise differentiable implementation of their fruit fly visual system model, and how they measure prediction accuracy in tests of their published constrained connectome DNM modeling method. That was also something we already looked at. I'm just mentioning it because that's something we took a closer look at here. Okay, the biophysical fidelity, this obviously could get very tricky when attempts at brain emulation begin to include plasticity. And you can notice that recent published work attempting functionalizations hasn't really gone there yet. And that's not even just there. For instance, in the years-long attempt to build a hippocampal prosthesis, changes in the system, plasticity, is also something that took a long time to get to and was particularly difficult. Okay, these last background literature points are about making appropriate use of your validation data. Machine learning tutorials might be a fairly good introduction. Again, I'll just mention the Neuromatch Academy. And then, yeah, I'm pointing out the Allen Brain Atlas and Blue Brain here as two examples where you might find existing data useful to developing validation protocols. So let's look at our developing of the metrics here. We're far from having validation metrics for every proposed success criterion, but let me walk through a possible development of one of them here to point out some considerations, just so you can see the sorts of things to think about, or an example of some of the things to think about. Take the success criterion P3 that was in our set of 8 remaining criteria. Observable behavior and responses are sufficiently similar. A little bit of reformulation has to be done there to make that success criterion apply to small in silico examples. Let's say that the I/O transfer function determines behavior and response to the system. And when we're working with in silico, we actually have everything available to us. We have this fully known ground truth system, which we purposely do, of course, on our challenge platform. And in that case, if we wanted to, we could explicitly write out the full transfer function as an equation. Now, that's where the first consideration comes in, because, yeah, even if that's possible, and if we could attempt to compare system transfer functions explicitly, that's not really something we want to do. After all, in whole brain emulation, we want to find a possible separation of scales that enables substrate independence for the functions relevant to our success criteria. In other words, we want it to be possible that the emulation involves implementation with somewhat different functional operators, and we purposely want to allow a degree of difference. So even in our in silico example on our platform, it's better to validate I/O behavior by using a battery of validation data. Another consideration is about how we want to measure I/O differences. So what matters downstream? For example, consider spatiotemporal patterns of activation in hippocampal CA3 and CA1 regions. Spiking probabilities there and time varying and the timing of those spikes vary significantly over successive repetitions of the same episodic memory task. So if you read some of the publications, for example, by Dong Song and others, the multi-input, multi-output hippocampal prosthetic model, it includes a validation process that compares both cognitive performance and the sub-threshold envelope of membrane potentials in an ensemble of neurons rather than focusing on spike trains. It's just one example of something to consider, even if you already know that you care about the I/O transfer function. And then, of course, we have to consider the size and composition of the validation test data. You've probably already noticed that developing a score with which to determine if one success criterion has been met might require a combination of metrics But on the other hand, in a specific circumstance, like when working with a small in silico example system, you might find that multiple success criteria can be judged using the same set of metrics. Oh, how wonderful. For example, our reformulated P3 criterion as well as N1 and N2. At present, the evaluation report that is produced when our challenge platform compares an attempted emulation with the original system, this is the set of tests that's applied, showing it here an example of that. The example here is of a fictional future brain reconstruction company, We Reconstruct LLC, that sent us their submission of their attempt, their method in February of 2025, pretty soon. Evaluating structure, neuron and synapse identification hits and misses are counted. We create a graph edit score. The graph edit score is a score that's produced by giving weighted scores to the number of edit operations of various types that you'd need to do to convert the structure of the submitted emulation model back to the structure of the original system. So that's by adding or removing cells, adding or removing synapses, changing cell or synapse type, those sorts of things. These are operations that you can then give some value or cost. To evaluate a meaningful function match, we use a validation stimulus set that tests an intended hidden function of a neural circuit embedded in the so-called ground truth system. I could get into that a bit more. This is really more detail of how we go about trying to aim what people who attempt to do an emulation are really getting done, like whether they're really finding something that you meant to find or would like them to find in the system. So I won't go into that too much right now. But having that there, having this intended function that allows us to test whether the emulation method used was able to discover the functional purpose of the original. We also run comparative sensitivity analysis using random input and compare the effect of spontaneous firing. That platform isn't ready for launch yet, and the evaluation metrics server still needs quite a bit of work. Okay, so this is the point where I'm going to check what the time is. Right. Okay, we're about halfway through our time, and we're also about halfway through this this set of associated topics. And I'm thinking that we could switch to some discussion right now. That might be a good idea. And in fact, we might take a brief 2-minute break. I could use one. And then, we'll see whether we go on to the next parts or not. There's actually a lot of interesting stuff about constraints and scale separation that I would love to talk about, but again, we don't have to do that today. So how about we take a 2-minute break and then we come back, and I'm just going to jump forward to a piece of discussion so that we can do that, and then we'll get back into other things if we have time. All right. Okay. See you in a minute or two, if you don't mind. And one more. There's a cat here who wants to be fed. And of course, because of the time right now, there are a bunch of AI otter note takers that are trying to join the session as well. I've let one or two in just so that we can get that record as well. That's probably very good for this discussion piece. So let me find where to jump forward to here. See, where were we here? I see, okay. At that point, this will be the first piece of discussion. All right, so an important part of what I'm trying to do, of course, is I'm trying not to just present, but I'm trying to get some input from you about what you think about all this. So I had prepared a few places. Oh, I should actually pop out of this so that I can take notes. Where I want to ask you some questions and then take a few notes about them. So let's see here. This is, I'd love to hear your thoughts on methods to determine success criteria. And I want to start by trying to seed the conversation with some ideas. So as you've already seen, you know, we did a survey and we also looked at a previous roadmap, but you could do a survey of a large group of neuroscientists and leading experts. You can, for example, also start by, say, surveying only those who are already clearly interested in whole brain emulation to ensure that the success criteria align on goals. Then you can publish success criteria to receive feedback and critique for improvement. You can distill the success criteria from literature by examining how results and methods appear to make progress on or be limited with respect to whole brain emulation goals. You can compare that, for instance, with the treatment that we gave the Lapalainen paper 2 weeks ago. And you could build a milestone timeline towards whole brain emulation and produce success criteria subsets for each of those milestones. Those are just some ideas, but I am hoping that some of you have already thought this through in greater detail and would love to contribute what you've been thinking should be done or have some other ideas input on this. Go ahead, Yunsuk.
All right. Thank you for the great summary and the presentation. So I had some, I guess, three things to say. One is that I guess all of the metrics that you have presented focus mostly on normal behavior, right? When things are functioning properly.
Mm-hmm.
So I think what's important, especially in terms of teasing out the causalities, it's important to check whether they, the system, the whole brain emulation, and the counterpart break in the same way. For example, if you freeze a part of the brain or disable as non-invasively and do the same on the brain emulation, would they behave or malfunction in the same way? So that's the first thought that I had.
Yep.
The second is, you already mentioned a little bit about this, about emergent behavior, right? So again, most of the stuff that you presented are based on what behaviors already exist, known behaviors. But what about certain situations which would require a totally new kind of behavior to emerge? And under that situation, with the brain emulation and the counterpart generate the same kind of emergent behavior. So this is more about like extrapolation rather than interpolation, right? And last bit is, I guess this gets on in the area of subjectivity. So how do you know that the whole brain emulation has feeling and so on? I guess this is very difficult to objectively measure. But one way, I guess I've said this sometime in the past during one of these workshops. One way is, let's say you have two hemispheres of your brain, and then let's say the whole brain emulation must have two hemispheres. So would it be possible to somehow link your the human's left hemisphere with the right hemisphere of the whole brain emulation and somehow switch on and off between the biological hemisphere and the emulated hemisphere. And you can actually click this switch to switch between the emulation and your biological brain and see if you notice anything different, right?
Mm-hmm. Yeah.
So yeah, I guess that'll be one way to figure this out.
Okay.
That's all I have to say.
Yep. No, those are great. As far as that subjective one's concerned, I mean, as you mentioned, obviously this is difficult to measure. It's also, that's one that you probably can only get to really once you're dealing with patient level things. And then, you're the experiment you're suggesting here assumes, for instance, that you still have a living human and you have emulation as well. So those are a few hurdles that I could see. And there's also the chance, of course, that if you determine that there are differences in the feeling, it might, there could be all sorts of reasons for it, but that's exactly what we want to find, of course. I just wonder at what point you start to see differences. And then, of course, there's the question of what differences are okay, the permissibility of it. That's this idea of an envelope of, I guess, healthy, the path that your healthy brain can take in terms of states or in terms of learning. It doesn't necessarily need to be identical between the original and an emulation, but has to fit within some envelope that is acceptable. I don't quite know how to express that yet, but that was another thought that came up. How about some more ideas?
I'm sorry. I have one more thing to say.
Yeah.
So I guess one really, really valuable aspect of whole brain emulation is that even though we cannot really understand consciousness using our biological brain, if we have a accurate whole brain emulation system, then the real benefit of that is that this whole brain emulation can actually study itself non-invasively to do all sorts of different experiments to test the subjectivity angle without hurting itself. Right. So for now, the only way to subjectively measure anything and make any scientific statement is for me to insert an electrode in my brain and so pass some current or do something to see or feel how my subjective whatever thing changes as I administer this current into my brain part, some brain part, right? And it's very limited. I don't want to kill myself doing so, but the whole brain emulation can actually do something like that to itself. So that's it.
Yep. Great point. Yes. Logan, were you trying to jump in just now?
Oh yeah, I just had an add-on to the subjective experiment that was suggested regarding a hemisphere, because I've thought about very similar things. I've actually thought about hemisphere-based, sort of on a tangent. I've thought about hemisphere replacement as a modality for gradual replacement uploading, except that it wouldn't really be quite as gradual, but I think that would be sufficient. But as a back on the topic of success criteria, I think that what was mentioned about using that as evidence, using sort of this prosthetic brain part as evidence for the success of sort of a subjective form of evidence, but evidence nonetheless for the success of an emulation or part of at least of part of the brain. I think that you could scale that down further to smaller parts of the brain, and that might be something that would be more feasible to do at a sooner time point in patients, probably in treatments for traumatic brain injury or something similar to that would be sort of the corollary.
So you mean things like, for example, let's just take the example of the hippocampal prosthesis because I'm very familiar with it. That could be seen as, oh, does this change your subjective experience in some way when you're turning that device on or not?
Right.
Now, there's some info there because, when they were conducting their experiments with patients who were undergoing surgery for epileptic to remove their pieces of brain because of epileptic seizures, they also asked them about their subjective experience in cases where this model that was stimulating and recording, if it was turned on and operating normally, operating as the model should, turned on, producing random output, or turned off, and whether they could tell the difference, not just in the context of the performance task, but just generally their sense of it. And of course, this wasn't a case where they scooped out the hippocampus and then put in a prosthetic. It was a support model, like it supported proper and memory encoding, or at least that was the idea. And the main outcome of that was simply that the patients had absolutely no idea when the model was on or off. They couldn't tell.
Well, that's a good sign,
Yeah.
I suppose. Yeah, and I think as more and more elaborate forms of that type of medical device come into being, especially as you alluded to with actual replacements for parts of the brain tissue that are missing, I think that we'll get more and more subjective evidence for the potential success, at least of the conscious awareness not being altered by being part of an emulation. So yeah, I've thought about that as possibly the only way to test the sort of subjective qualia aspects would be to do something like that, and that seems like beyond all the sort of behavioral outcomes, the subjective experience would have to be tested in some way similar to what Yunsuk suggested.
Yeah, I think there are quite a few people thinking about it that way. I know that one of our other team members, Arseny, who may also be here, has been thinking about a very similar approach for testing the effect on subjective awareness. Yeah.
I've been very curious. Sorry, this is again is slightly a tangent, but I've been very curious about the idea of a early attempts at a corpus callosum based like interfaces, like where people would actually try to um, take the information passing between the hemispheres and record from that in a fairly high density fashion. If anyone knows about that, feel free to reach out. But again, that's more of a technical question than a success criteria question. So I'm not sure if this is relevant to the actual discussion here.
Well, maybe somebody knows. Let's
Yeah.
see if anyone wants to chime in on that. Maybe not.
All right. Well, we'll see. We'll see if someone finds out.
So I'm not sure if everyone who's here can actually just speak through their microphone, which makes things easier. So I'm noticing that there has been some discussion in chat, and I'm not able to really pay attention to it because there's already too much going on my end, but I'm hoping that I'll be able to do afterwards. I do see here that Michael asked, what was the article by Conrad? And right now, I can't quite remember what that was referring to. Which one what was the subject matter I was mentioning? Not sure if you can speak, but if you can, maybe remind me.
Yeah, so article, I think that was for, actually, let me look back through my notes. Yeah, see exactly where it was. I'll get back to you.
Do you remember, like, I could go through the slides if it helps you get there, but anyway, I'm maybe we can just, oh, Ken, yes, go on. Oh, you had your hand up a second ago, but then there was no.
I'm sorry. So I didn't have anything relevant to the Conrad article. I had a separate question.
Yeah, sure.
So you probably covered this, but the success criteria from the emulation's own point of view, comparing to its memory. So, for instance, if I went in for brain surgery,
yeah,
and I came out of that brain surgery, and there were obvious deficits, and I could look at those deficits and say, oh, I used to have color vision, but now I don't have any color vision, right? Or I used to be able to see the right hemisphere of my visual field, and I don't care because it seemingly doesn't exist, but I'm not bothered by it.
Yeah.
So what I'm wondering is there's probably a whole literature on success criteria in brain in strokes, etc., and how relevant from the internal point of view of the patient. And I'm wondering how much of that could be transferred over to a brain emulation criteria.
Great. I love this. This is another source we can go and dive into to see if that's there. Yeah, that would be fantastic. I hadn't really covered that. I think all I did so far, I mean, we can dive into the success criteria we collected. That's actually the next, say, discussion bit, not this one, but the next one, and see what was already there. But one thing was that I was focusing right now more on the objectively measurable, I guess, criteria or metrics for those. Subjective is relevant, very relevant, and of course applies heavily when patients are involved. Yeah, I've been a little distracted by the in silico stuff at the moment. That probably has me a bit off on a tangent there.
And I also feel like it gets a little bit to the question of qualia, because I mean, you tell me not, but if I'm an emulation, let's say, and I say the ice cream that I'm tasting tastes the same as it used to taste,
right?
I consider that a relevant to the qualia. It's theoretically, in some in some really theoretical case, you could be misremembering, but this is a qualia to qualia. Yes, going through a computational process, but it's a qualia to qualia comparison, and that, I think that's about as close as we can ever get to really to saying this subjective comparison to past memory of qualia.
Yeah, that's a good test. So going a bit beyond just checking if, say, a survey of memories that a patient decided they wanted to check was available, and testing for these kinds of experiences and sensations, and qualia. Yeah, that seems quite relevant. Now we can.
I'll just go ahead and say it, though.
Yes. Yeah.
The philosophical zombie argument is that you would self-report that you are experiencing the qualia, but you're not.
Yeah. Okay. You said I'm just gonna say it. That's probably because you're already thinking about all the I mean, it's complaints you can have about that whole scenario. But yeah, you're right. I mean, theoretically, if there is such a thing as philosophical zombies in a case where you've actually done an emulation and not just made a big, lookup table and have a computer run through that, then it's, I mean, if there were such a possibility anyway, if you could still get philosophical zombies, then in theory, a person might report that they have the same qualia, and yet no one's home. Yeah.
Isn't that just why you would need something like a hemisphere replacement first, so that you could?
How can you tell? What if the first hemisphere replacement already turns you into philosophical zombie, and you're still responding as you normally would?
And that's why you need it to be reversible. You convert it one way, you convert it back the other way, and then you allow the initial entity to compare. Was it there or was it not there?
Who's comparing? I mean, the philosophical zombie doesn't realize that it doesn't have true conscious awareness, subjective awareness. So you're turning into philosophical zombie, then you're turning back into normal. How do you know that you would be able to tell that you were briefly not yourself?
Yeah,
especially going through memory, and that memory could be false. I mean, Daniel Dennett covered this quite a bit in Consciousness Explained.
Yeah. Dennett's one of the ones who does the best job of explaining that there really is no way out of this. What you have to do is pick your model and decide that that's how you're going to model the world, and you're done with it, which I'm okay with. But that is the we will run into that counter argument forever because it actually is sort of non-empirically solvable.
Yeah, to a degree. I don't think that all of these things are quite as unsolvable as some philosophers will say, or some neuroscientists will even say. I think that as you start to understand, say, the causal chain of what's going on, you can sort of see where things are being put into a temporary buffer, and you therefore realize you were just aware of this and all that. It can become more experientially understandable why something would feel a certain way and why there's that connection. And I do believe that that can then eventually make this problem go away. That's just my personal belief, but I guess, you know, it depends on what you think. Whether scientific explanation is ever able to change people's deep felt feelings about things. Who's this? Oh, I see. Arseny, did you put your hand up first? I think you did.
Yep. I just wanted to reply about that. You might not know that you were a philosophical zombie for a moment. I know that's my idea of protocol still involves a conjecture that you'll be able to discern that, but that's as small a conjecture as I can imagine. It's definitely, I think it's definitely better than listening to the entities that might be at the moment of saying to be philosophical zombie at that very moment. It's better to listen to something that is reverted back to the initial condition, and at least to not have the undecidedness about what is talking to you right now.
Yeah. I mean, to some degree, you can do studies like that, maybe not by replacing part of a brain with an emulation, but you can at least silence neurons in some part of the brain chemically for a while and then have them come back on. And those studies, we could definitely get into that literature. And
yeah, and if you can silence the neurons in some part and you also have that part emulated, then you can kind of replace it temporarily.
Yeah, depending on brain-computer interfaces and all that. Yeah.
Well,
there's a technological hurdle, but there's a technological hurdle in all of this, so that's fine. Yeah. Yunsuk.
Right. So I wanted to mention one other thing. So in your treating whole brain emulation as a machine learning problem, the first I guess assumption is cognition is basically information processing. So I guess we need to define what information is. Is it in the Shannon sense or what? And I think that the brain may be processing information, but it could be a different kind of information from what we are pretty much used to, right?
Yeah.
So I guess that's another thing to consider.
I'm not 100% sure what you're going towards. Can you give an example of something where, say, the way the brain might be processing information is not something comparable with the, treating cognition as an information process or treating it as machine learning in some sense, or similar to in some way? Where would
I can elaborate on that a little bit. So if you look at Shannon's papers, when he says information, in his view, information does not have any meaning to it.
Yep.
Okay, but in the brain, I guess this is kind of related to the current debate about large language models, whether they understand what they're saying and so on. So in one sense, in the Shannon sense, they're basically processing information very efficiently. But by saying that or claiming that large language models do not understand, we're saying that the information that they're processing does not have the same kind of meaning that humans do.
Well, so I've already put on the notes here my immediate response, which is possibly wrong, but this is how I see it. I don't think that patterns of activity in the brain as such have any meaning at all. They can't unless they're in the context of something else. So it's the downstream processing. So if you're using, say, some attractor to store a piece of memory, it only has meaning in the sense that you can decode it and then use it somewhere else and produce a response, or put something in working memory and then realize, oh, I was just aware of this, that sort of thing. So I think that meaning of information really only exists in a context, the same way as putting letters on a piece of paper those are just bits of ink. They only have meaning in the context of a language that we're using to communicate, things like that. So
yeah, that's very true. So information, if you think of it as already containing some meaning to it, I guess that's very wrong. And what matters is how that piece of information is being used, maybe in the Wittgensteinian way, right?
Yeah,
so the meaning of a word is not in what it indicates in the environment, is rather in the usage in the language environment.
absolutely. In German, the word gift means poison. So there you go.
Oh, right.
Okay, Logan.
I just had it's sort of a different point, but I had a kind of a meta point about something you mentioned earlier, which was robustness.
Yeah.
Success criteria, and what it brought to mind for me was that, I mean, personally, I think that, you know, the sort of ultimate goal of human whole brain emulation will take some time, and we don't know exactly what research directions are going to evolve over that time period. And so I think having um, some level of flexibility may increase the robustness of this type of success criteria. So, for example, in connectomics, there may be different experimental techniques that are used for different scales. So, I mean, we're seeing a lot of EM used now, but there may be some light sheet microscopy that, with expansion, that might be used in more so in the future. And, you know, other things may still arise even beyond that. And so, and at the same time, many of these things may evolve in parallel. So probably EM and light sheet expansion microscopy are going to be used, are going to be developing in parallel for some time yet still. And there may be other competitors and so on and so forth. And so I almost wonder whether we should, or I guess my point is that we should just be aware that it may be valuable to accept that different directions that may seem tangential can still help get us to the goal. And so having success criteria that are very rigid that say, oh no, it has to be exactly this type of research in order to get to whole brain emulation, I think that can be somewhat constraining. And even beyond that, the larger sort of community of researchers who may or may not be interested in whole brain emulation are all going to be contributing to things, or well, at least some of them may be contributing to things that may be helpful for whole brain emulation. And so I think having sort of an acceptance of the possibility of unexpected things coming in from outside of the mainstay of the field is also very important to be aware of when outlining these criteria.
Okay, that's a good point. Yeah, so I did kind of touch on that a little bit in the sense that the whole project of trying to define success criteria is an iterative thing, and it needs to be informed as we go along. But you're absolutely right that that's something that should be emphasized, and that there are some things you can do to make yourself less rigid. Yeah.
Right.
Emmanuel.
Yeah, can you hear me?
Definitely, yes.
Yeah, okay. Yeah, exactly. What Logan said is interesting, because probably the first thing we're gonna have to evaluate is gonna be like those big lo-fi. You didn't talk a lot about it, but this lo-fi maybe MRI driven or attempts, they're gonna arrive quite soon with ML, and probably people claiming they reached like whole brain emulation, and we'll have to test the success criteria on these in the first place. That's really interesting. So I think, of course, it's not going to be enough, those like MRI approaches, but that would be a good test, if to be able to say, well, it doesn't work, or it's not enough, right? So
yeah, that's a good point.
Maybe you have to develop tools specifically for this.
Right. That's a very good point. So in addition to having to look at things like, for example, the Lapalainen paper we went through 2 weeks ago and see where does that line up with where we would be going, and where can you do this kind of stereotyping and all that, it's true that, yes, these lo-fi things are going to be publishing papers and making claims, and you need to be able to test how does that compare with where we're trying to go I absolutely agree. Yes, you're right that I didn't mention it a lot yet. We're halfway through the slides that I had, and there are topics coming up about constraints and about scale separation, and then there's discussion about lo-fi as well as part of that. We may not be able to get to that today unless we all want to stick around much longer. I'm ready for it, but I don't know who else is. I have a few more things, though, about success criteria and metrics that maybe we can talk about as well, unless there are still further ideas, thoughts about this one, about how to determine success criteria. If not, then I'll get us into our second piece of discussion about that part of, I guess, of this whole, I guess we can call it a chapter of the roadmap or something that we're working on. Okay, so critiques of and suggestions for success criteria. I think we did a little bit of that above already, but maybe not directly related to the work that we had put here. I want to make sure to put, let's see if I can grab that from here. I want to put the URL for that spreadsheet in there for you. I'm going to copy it to the chat if it'll open for me. Here we go. Putting this in chat now. Let me know if this works or if I somehow mangled the, I want to make sure everybody can see it. There. I might have mangled the URL and then it might not open, but we'll see if that works. Logan, yeah, I see what you just said here. Yeah, if you have to go, you got to go. Sorry to hear that. Maybe that's a good reason not to do everything today. We'll see about that in a second.
Yeah, I was just wondering because I wasn't sure if there was something specific that you needed me for with regards to the roadmap group, and communicating things from that. But
it's mostly just to have your input on these topics, so you don't have to worry about that. Yeah, I don't need anything from you that describes the roadmap project or anything like that at this point.
All right. Well, yeah. Yep. Thanks for having me join this, and yeah, I'll see you around. But yeah, I look forward to hearing more from anyone. And
okay, great. Have a good one. And yes, I see Yunsuk that you're saying you have access. That's very good, because the like the next question that I have here is, are the proposed success criteria in there? Are there some that really don't make sense to you, appear faulty? Are there success criteria that make a trade-off that we should be aware of? Should they be stated in some other way? Are there criteria that appear to be missing? So basically, your take on, or whatever you want to say about this spreadsheet at the moment. I realize that maybe this is a difficult thing to do here online because there's a lot to look at. There's a lot to read through in that spreadsheet. But yeah, let's see if someone's interested in doing that before we move on to something else. And I guess I'll take that as an opportunity to step away for a moment because everyone gets a chance to take a look at that spreadsheet. Also, feel absolutely free to comment directly on the spreadsheet, and you can do that now or later. That's all fine by me. But if there are things that you think are worth discussing now, you don't have to rush. still take your time reading, and I'd be super interested in that. Let me see if I can get to where I can take some notes on it. All right. Thanks for being here, Daniel. Someone put their hand up, I think, and I can't see who. Ah, Emmanuel.
Yeah, it's me. Just I was just what Kenneth said about the taste of the ice cream.
Yeah.
Made me think about what like maybe the less resource intensive way to test, like, because the taste is quite interesting. Either test vision or, like maybe speech, but you have to find maybe the thing that is the easiest to test. The taste is quite good because it's not a lot of receptors involved. So maybe not. That's just like maybe something we could think about. Like, what's the best shortcut, to.
Okay. Yeah.
And you also have to retrieve information, so to be able to hear the model or the humiliated person say
Yeah.
how it tastes. So that's kind of difficult, actually, but it's an interesting.
Yeah, it is. Yeah. Maria, I don't know if you can speak, but I think you might have said that you couldn't. Oh no, that was someone else. But is this directly related to what Emmanuel was just talking about? The link you just posted?
Oh, it's just generally relevant to the consciousness-related criteria. It's basically some work on taking different theories of consciousness and finding what criteria for consciousness each one suggests, and then examining the extent to which different AI systems may possess those properties.
I see. So it's a survey of sorts.
Yeah.
Okay. Yeah. Cool. I will tuck that into our Zotero. Yeah.
Just a quick comment about taste. Taste might actually be quite more complicated, because that the way that we remember taste is also strongly influenced by olfaction. So if you just have the taste like sweet, sour, all that, I guess that doesn't really fully describe our memory about taste.
Yeah. Okay, that's right. Yeah. Yeah, I mean, there are, and we're still talking about success criteria here that are specifically and purposely subjective, right? So then it's not a question of, is, say, a visual stimulus being processed correctly and coming out the other end of the lower visual system in the right form. So it's not something like that. Yeah. I actually didn't anticipate that, as we were discussing this, that there would be quite as many concerns about subjective awareness, and I'm not sure why I didn't anticipate that. So that's an interesting question. Yeah. Do a little bit of self-checking there. Does anyone else have any comments on what you're seeing on that spreadsheet at this moment? Maybe, you know, I mean, as I said, you can also put information there later.
Sorry, I'm the one who opened that whole can of worms.
Oh, don't worry about it. No, it's a good thing. I mean, obviously, checking my biases is for what I pay more attention to or less. It's just that, you know, because we're trying to get from success criteria to metrics, you know, it seemed like an obvious thing that if we're going to be dealing with smaller systems first, like animals and simulated systems and all that sort of thing, that we'd be looking for something more objective rather than subjective first. So that kind of got my attention first way around.
I mean, more to the point, if we're going to do any initial evaluation on non-human, then we can just kind of compartmentalize the entire self-report discussion away. We can just say, that's for another day, but what we're talking about here is neurophysiological measures, and that's just, you know, that is where we are sequestering and siloing our investigation for now, and we'll ask the subjective questions in a different venue or something.
You can appreciate that. You can, I mean, you can do that, but I appreciate sort of this reminder to check my bias here and be careful, because I mean, one of the things that I was trying to say earlier on in the presentation is that the success criteria that you initially choose or that you focus on and improve as you're working on a new field has a tendency to direct where it goes and where advances are made. And so if you focus completely on, say, neurophysiological measurements, it could be that you end up building a wonderful toolkit, but that it doesn't actually ultimately help you get to what we really want as a goal. So there's some important checking to be done there. And I think that's also what others like Arseny have expressed, for example. Yeah.
So on that note, I guess if you look at the brain rewiring experiments by Mirgan Kasur and his colleagues, we might be able to develop some kind of a test for subjective qualia by training animals. Like they trained ferrets to basically behave, whether this feels like vision or whether this feels like sound, with the rewired ferret brain. So they plugged in the optic nerve into the MGN, so that the left hemisphere, the auditory cortex was receiving visual input. So when you flash the light and stimulate auditory cortex, does it feel like a flash of light, or does it sounds like a ding? So I have to look at this paper again, but I guess there is some possibility of teasing that kind of thing out in both strokes, right, using animal experiments. So one other question regarding these columns. This is a question to Randall.
Yeah.
So I was trying to see whether I could suggest some other column, other category. And I was wondering how you came up with these different columns. Is objective? Is it objective, measurable?
It was a bit of a development. I mean, for instance, on the first sheet, you can see that we had a number of other columns, how we were trying to categorize and understand what belongs where and how to use it. What ultimately happened is that for this topically sorted thing, as we continued moving along, there were some that seemed really relevant anyway, no matter what, can you imagine an objective or way to measure something? Is it something non-technical? So we should put it in a totally different kind of category, different group. But really what happened, and I tried to point this out in one of my slides at least, is that once we had these, we also have this project going on for that brain emulation challenge where we're trying to use fully known ground truths as a way to help develop methods. And we wanted to see what could we apply to the kinds of examples that we could feasibly do there. And so it had to get down to something that is really only a neural circuit, not an entire organism necessarily, unless it's a really tiny organism, fairly small, and also aimed at this main problem of functionalization that we really wanted to make progress on. So that's why these particular columns popped up. But I can imagine that we might categorize rather differently for other reasons.
So do you anticipate more columns to appear based on our discussion?
Yes. Yeah, I would hope so. Yeah.
Okay. I'll think about it.
Okay, great. Anyone have any more thoughts about this right away? I mean, no problem if not. You can always do so later. Okay, if not, then I'm going to try one more thing, because if we can do one more bit of discussion now, then we'll have really done half of this whole thing, or at least as it was planned originally. Moving forward here. Okay, this is the discussion of how one might derive metrics. And again, I wanted to seed that conversation beyond the slides that I already showed earlier about all the thoughts that go into that, the considerations about how to derive a good metric, just seeding a bit here. So one thing you can do, of course, is as you start thinking about a success criterion and how you might measure it, you might just think about how you can actually formulate that not as words, but as variables. You can consider the scope of the criterion. You can consider possible measurements that are affected by those variables that you came up with in your formulation. You can consider a validation protocol, thresholds and significance. You can consider the data set that you would need, its breadth and length, and what that means about its significance, about the coverage and its limitations. You could consider how feasible a particular approach is in the context of these different milestones. So for instance, those in silico studies or fully controlled experimental arrangements in vitro, existing data sets that we have like the fly brain, and small animal studies, human studies. And then ultimately, there are probably some success criteria that where even the metrics for it are really only relevant to, a very large scale approach where the thought is about, okay, how do you do this with a whole population of patients? You can examine the validation protocols that are currently used in the literature with all of that in mind, and you can compare validation metrics or validation data as they do in AI and machine learning. So that's just to seed the conversation a little bit, but I'm very interested in the derivation of metrics that can help you determine whether a success criterion has been met or not, where of course I assume that the kind that you use with neural circuits, those kinds of success criteria, it's much easier to develop metrics for those than some others, especially if you're dealing with animals instead of humans, because they won't necessarily respond quite the same way. Yeah, thoughts? Or pointers to good literature, or what you use in your thinking about developing whole brain emulation or something related to that. Yeah, how do you develop your metrics? How do you define them so that you know they're aiming at the right thing, and also so that you can somehow guarantee that you really are still aiming at those success criteria, that you're not optimizing just for a metric that maybe ends up being a dead end.
I guess aside from normal function and function on the some kind of destructive condition, we can also think about peculiar behaviors like illusions.
Mm-hmm.
So would the whole brain emulation experience the same kind of illusion when they present them with it?
Yeah, those can be really useful. Yeah, so they point to mechanistic causes. So yeah, that's a great one. And then, of course, we could be talking not just about visual illusions, but illusions such as the sense of self, just to name one.
Exactly like the robot arm and tickling and stabbing.
Yeah.
And not just vision, but multimodal, like sound plus vision.
Cool. Yeah. Any others? If not, not a problem. And we might always get back to this. Then I have a different question for everybody. And I think maybe raising hands is a good way to determine this. I would like to get some understanding of whether there are any people who would stick around for more today or not, because if that's not a large enough group, then I think it's clear that we have to split this up into two parts. So if anyone here would like to just carry on to get into constraints, scale separation, and then discussion of the lo-fi thing, please raise your hand and I'll try to see what proportion of us that might be. Wow, I'm not seeing any hands. Yeah, Emmanuel would like to go on. Okay. And Maria. I am very proud of your persistence, your stamina, your strength. Great people.
Randall, there's some feedback in the chat to follow too.
I see. Most people are unavailable. Okay, so some people would be okay with sticking around, but since most people don't want to, yeah. Okay, so I think it's probably unfair if we, have like a group of 3 or 4 people carrying on, and everybody else has to just watch the recording. So it makes sense for us to split this in 2 parts. And as I mentioned, that means 2 weeks from now, because during the following week I'll be doing a lot of traveling, and yeah, but I look forward to it. And this also gives us an opportunity, of course, to just think through this stuff. And I will share the slides with you so you can get a head start. You can look at what's there, and then you'll already have all your ideas as we try to move through the next part. Great. Thanks, everybody. Very glad that there were some good ideas that I hadn't even thought of at all, and that none of us in the roadmap project had really thought about carefully, and that my biases were kind of put in check. That's also wonderful. Yeah, thanks. This was productive. Thank you very much.
Thanks, Randall. Bye, everyone.
All right. See you all again in 2 weeks. Bye-bye. And I'm going to stick around for you, Richard, so we can make sure the recording is properly done, or that we have one.
Yep.
Yeah, it's there.